Welcome to Veterinary Physiology lectures. Today we will learn the formation of ovarian follicles. It is called follicular genesis. Do you know that the growth of female germ cell involves a lot of biochemical interaction or crosstalk between and within the granulosa and theca cells? Oocyte grows in a follicle, supported and protected by granulosa cell. The successful growth and maturation of the oocyte depends upon the hypothalamo-pituitary gonadal axis. The oocyte develops in a follicle. The follicle is like a home for the oocyte. The oocyte grows, develops, and gets equipped within the follicle, and then it is released from the follicle. Let us understand the follicular genesis with a story. Once upon a time, there was a happy family. The little child in the family makes the home joyful. The parents love their only daughter and provide all the care, support for her future. It is not an easy thing for the parents because it involves a lot of hard work. The father goes out to work to earn money. There is a lot of expenses, for example, the daughter's education, tuition fee, not even mentioning other expenses for the management of the house. The parents do all the necessary for the welfare of their daughter. The father earns money and hand over to her mother, who manage the house properly. The parents want their daughter to be excellent in all things. The parents' desire is that their daughter should stand in her own feet, get married, and build her own home and family and live happily. There is always a heart-touch cry at the time of marriage for their daughter's farewell. But at the same time, they are happy because their daughter is going to make her own family. This is the story. Now let us compare this story with our topic, that is follicular genesis. The oocyte grows and develops in an ovarian follicle. The follicle is a home for the oocyte. The granulosa cell take care of the oocyte like a mother. The theca cell is like a father. The theca cells produce androgen. Then the androgen is converted to estrogen by the granulosa cells. Many more substances are synthesized and secreted by the granulosa cell for the developing oocyte. The granulosa cells are closely contact with the oocyte. As the father earns for the family and mother manage the home, support the child, and make all the things available for her necessities, similarly, granulosa and theca cells do the same to the oocyte. Theca cell layer have the blood vessels, that is, blood supply is present in the theca layer, but it is absent in the granulosa layers. Therefore, the granulosa cell layers does not have the blood vessels. The theca cell produce androgen under the influence of LH. Then the granulosa cell convert the androgen into estrogen under the influence of FSH. I want to focus here three important points to understand the follicular genesis. The first point is recruitment of the follicles. Second is gonadotrophin independent growing follicle. And the third is the growth of the follicle dependent on gonadotrophin, that is FSH and LH. Before going into discussion of follicular genesis, let us first understand the formation of primordial follicle. The primordial germ cell have to migrate to the genital ridge, increase their number by mitosis division. Now it is called oogonia. Oogonia give rise to the female germ cell, that is oocyte. Oogonia is also known as the stem cell of the ovary. The rete ovary, that is the counterpart of rete testis, secretes meiosis initiating factors which cause the cell to undergo meiosis, that is reduction division, where the chromosome number becomes half. Oogonia transform to primary oocyte, enter into meiosis division, but arrested at prophase 1 at dictyade stage. Why? Because the follicular cells which surrounds the oocyte increase their number and they secrete meiosis inhibiting factors that inhibit the meiosis division. The follicular cell prevent the developing oocyte to complete its first meiosis division. Therefore, it is arrested at prophase 1 at dictyade stage. Now, this follicle is called primordial follicle. It is a resting follicle. This primordial follicle remain in the ovary for years. Now, let us see the three important points of follicular genesis. The first point is the recruitment of the ovarian follicle. Some of the primordial follicle is recruited by the intra-ovarian factors for growth and development in every cycle, that is estrous cycle or menstrual cycle. The intra-ovarian factors, that is the growth factors or other hormone-like substances in the ovary. The number of recruitment of the primordial follicle for follicular genesis is also under genetic control. We know that the primordial follicle is a resting follicle in the ovary. When they are recruited by the intra-ovarian factors, it is awakened from their sleep. Not all the primordial follicle is secreted at a time, but few numbers of primordial follicles are selected by the intra-ovarian factors for growth and development. These awakened follicles start growing and finally become mature and form the Graafian follicle, which will then ovulate. The primordial follicles are awakened by the intra-ovarian factors and then they start growing, that is, enter into growing phase. Let us divide the growing phase into two stages. First is gonadotrophin independent follicular growth. Second is gonadotrophin dependent follicular growth, that is, follicle required FSH and LH for their growth and maturation. Gonadotrophin independent follicular growth. The oocyte is very active during initial period of follicular genesis. There is intense synthetic activity in the oocyte, synthesis of large amount of ribonucleic acid. Though the oocyte is active metabolically but is still at dictyate stage, it is still in arrested stage, arrested at prophase 1 of first meiosis division. What about the follicular cells then? The follicular cells become cuboidal from flat follicular cells. Increase their number by mitosis division around the oocyte. Then the follicular cell delineated themselves by forming an outer basement membrane, that is called membrana propria. Another boundary layer is formed by the secretory product of both follicular cell and the developing oocyte. This layer is called zona pellucida. The zona pellucida lies within the granulosa cell that immediately surrounds the oocyte. How? Let us see it. The granulosa cell which are immediately surrounds the oocyte form the cytoplasmic projection, contact the oocyte through the zona pellucida. It plays significant role for providing nourishment to the developing oocyte, like a mother caring for the child. Therefore, the zona pellucida lies within the granulosa that immediately surround the oocyte. Organization of granulosa and theca cells. Now the follicular cells that surround the oocyte are called granulosa cells. They are very active metabolically, increase their number by mitosis division, forming several layers around the developing oocyte. The theca cells organize around the basement membrane, forming theca interna and theca externa. Theca layers have the privilege of having blood vessels or blood supply, but the blood vessels are absent in the granulosa cell layers. Therefore, the granulosa does not have the direct contact to blood capillaries. This is the growth of the follicle without gonadotrophin or independent of gonadotrophin. Here the important significant point is that the follicle is unable to form the antrum, that is fluid-filled cavity within the follicle. The antrum is absent in gonadotrophin independent follicle. Therefore, gonadotrophin independent follicle is also called pre-antral follicle. Gonadotrophin dependent follicular growth. When we say gonadotrophin dependent follicular growth, then the theca and granulosa cells must develop receptor for gonadotrophins, that is FSH and LH. As the granulosa cell develop receptor for FSH, the theca cell develop receptor for LH. Interestingly, here we can see two-cell mechanism. What is this two-cell mechanism? There is involvement of two type of cell for one specific goal. The theca cell produce androgen under the influence of LH. The androgen diffuse to the granulosa cell layers, where androgen is converted to estrogen by the granulosa cells. This magic is done by the enzyme, that is aromatase. This process is called aromatization. The granulosa cell under the influence of FSH convert the androgen to estrogen by aromatase activity. The FSH also stimulates the granulosa cells for the secretion of fluid. The fluid is also known as liquor folliculi. Therefore, the fluid accumulates in the follicle. Many of the small fluid-filled space coalescence to form the antrum, that is, large fluid-filled cavity. Therefore, the antrum formation is a clear indication of gonadotrophin dependent follicle. Formation of Graafian follicle. The estrogen production increases due to the action of FSH on granulosa cell. Estrogen stimulates the granulosa cell to increase their number by mitosis division. Therefore, the layers of granulosa cell increase due to the stimulatory action of estrogen. The granulosa cells are stimulated by its own secretory product, that is estrogen, self-stimulatory. The follicular fluid also increases its amount. The follicular size increases significantly. These large follicles can survive with little amount of FSH due to the development of a greater number of FSH receptors on granulosa cell. This follicle is now called dominant follicle. The granulosa cells of dominant follicles produce higher amount of inhibin hormone that inhibit the FSH release from anterior pituitary. The high concentration of estrogen also has negative feedback on FSH release. Therefore, the FSH concentration decreases. The growing follicles which are still dependent on FSH for further growth and development and maturation undergo atresia due to the decreased concentration of FSH. Atresia is the degeneration of follicle, whereas the dominant follicle can survive due to the greater number of FSH receptor on granulosa cell. So the little amount of FSH is sufficient for their growth, survival and functioning. The dominant follicle or follicles which are going to ovulate is called Graafian follicle. This is fully matured follicle. The formation of Graafian follicle or antral follicle is fully hormonal dependent. Hormonal regulation of follicular genesis. Now we know that the early follicular genesis or initial development of follicle is independent of gonadotrophin, that is FSH and LH. We can also say that the formation of preantral follicle is independent of gonadotrophin. But the gonadotrophin dependent follicle desperately needs FSH and LH for their growth, development and maturity. So the hypothalamus, pituitary and ovary have to work in coordination. The hypothalamus produces and releases gonadotrophin releasing hormone, that is GnRH. It is a protein hormone. It has to be transported to the anterior pituitary through portal circulation. GnRH stimulates the gonadotropes for the synthesis and release of FSH and LH. FSH act upon the follicle for growth and development, stimulate the granulosa cell for secretion of fluid, that is liquor folliculi. This led to the formation of antrum. LH act upon theca cells for the production of androgen. Androgen pass through the basement membrane and converted to estrogen by the aromatase activity. FSH enhances the aromatase activity in the granulosa cells. Therefore, the estrogen concentration increases significantly. The higher concentration of estrogen is become negative feedback to hypothalamus and pituitary. However, the low concentration of estrogen is positive feedback to hypothalamus and pituitary. The granulosa cells of dominant follicle produces higher concentration of inhibin that prevent the FSH release from the anterior pituitary. Here we can see the positive and negative feedback mechanism of estrogen. Importance of luteinizing hormone, that is LH, for follicle maturation. The LH surge before ovulation is important for maturation of follicle. We know that the oocyte is arrested at prophase 1 due to the meiosis inhibiting factors. The developing follicle possesses the primary oocyte until the LH surge. When there is LH surge, it causes the resumption of meiosis 1. Secondary oocyte and polar body is formed after completion of meiosis 1. Then the secondary oocyte enter into meiosis 2 but arrested at metaphase 2. At the time of ovulation, the oocyte is secondary oocyte arrested at metaphase 2 in most of the mammals except few like mare and bitch. LH surge also luteinized some of the granulosa cells, caused them to produce progesterone instead of estrogen. So at the time of ovulation, many of the granulosa cells producing progesterone instead of estrogen. Conclusion. Let us conclude with some points. I have mentioned many points in this lecture. Let us conclude with few points. First, recruitment of primordial follicle. It is by intra-ovarian factors. Second is the gonadotrophin independent follicular growth. Third is gonadotrophin dependent follicular growth, that is formation of antral follicle. Fourth is fully maturation of follicle required LH hormone. LH surge convert the primary oocyte to secondary oocyte. Fifth, the luteinization of granulosa cells for the production of progesterone before ovulation. What is the purpose of follicular genesis? Do you remember the story I told you about a family and their daughter? The main purpose of the parents is to equip their daughter, make her fit to stand in her own feet. Similarly, the main purpose of follicular genesis is to make the oocyte fully equipped for successful fertilization, embryogenesis, and finally to form a healthy individual. Thank you for watching my videos. If you have any suggestion, you are most welcome.